before we get started today, we have a guest, Dana from the Academic Success Center wants to just talk to you for a few minutes about all the services and workshops and things like that I have going on down there. So I think I probably need to somehow let you share the screen, right? I'm just trying to remember how to do that.
Yes, you're going to add me as a co-host, I believe.
Okay. Okay, so I see that in okay, so what I'm going to do is I'm going to unshare mine and I'm going to allow the participant to share screen. So I think that will let you do so. So I'll just stop my share and that should work. So give it a try.
All right. Okay, yeah. I think you can share my screen here. Right. Can you guys see the PDF?
I can see it.
Okay, perfect. All right. Well, I'll go ahead now. I'll just remove my video just to help with my internet connection here. Okay, perfect. All right. Well, hello, everyone. Again, my name is Dana, and I'm from the Academic Success Center, or ASC. Hopefully, you guys are familiar with us already. I do see some familiar names here right now, but for those of you guys who doesn't know who we are formerly known as the Skills Center. We just rebranded to the Academic Success Center, and we do offer free tutoring and study support to all Keanu College students in any program. So we are available to support students in a wide range of subjects, including science, math, writing, and English. So the ASC consists of 3 academic content specialists and an academic success coach. So Kostav is our math and engineering specialist. He can help you with all courses related to math and engineering, such as calculus, algebra, traits, stats, physics, and so on. And of course, he can clarify any math concepts and help you in any math problems, as well as just to develop the numeracy skills that you guys would need to be successful on those courses. Next, we have Mindy. So she's our English, writing, and humanities specialist. So she can help you guys understand different concepts in English and humanities, also with a variety of courses such as sociology, English, philosophy, and more. And of course, to also assist you with essay or thesis writing, grammar and language help, proofreading, and so on. And then there's me. Again, my name is Dana, and I'm the science specialist, and I can help you guys in all things science, such as your course right now in bio. If you guys have any chemistry courses, any microbio, ENVT, anatomy, etc. And I can also help students in social sciences like psychology, sociology, and so on. So yeah, I can assist you guys in content review, laboratory reports, research projects, and more. And last but not the least, we have Claire. She's our academic success coach. So her role is to provide support and resources to you guys to achieve academic success. So if you guys need help with time management, organization, goal setting, note taking, or any other study strategies, she's the person to go to. Claire also works with the register and meet students on the RTW list. We have individual Keanu emails, but just to simplify things, you guys can always email us at our general email. So it's right here, academic.success@keanu.ca. So this basically just contacts all of us so we can just talk amongst ourselves which person is the best to contact you and work with you. We also do have a Facebook page at the bottom here. Please do like and follow us as we post a lot of our updates and events in the center or even just in Keanu in general. So as seen here, we are open and available Mondays to Fridays from 8:30 a.m. to 4:30 p.m. And we are expecting to offer extended hours for math and trades and possibly science up to 6 p.m. in the upcoming weeks. So yeah, we will be posting that on our Facebook. So yeah, feel free to follow along. Our center is also usually a drop-in center for those of you who haven't come in our center, meaning you guys can just come individually or as a group and study there, use your computers or printers, even book our study room if you need it, and just come to any of us when you need help or when you're stuck on something. But because of the situation we're in right now, we're just all currently working from home. So the center is temporarily closed. We are still available, but you guys are just going to need to email us and set appointments with us for any assistance or tutoring help that you need. So just by saying that, we're always here to support you guys, but we also do have some boundaries. So we still expect students to, of course, attend classes and make use of your instructor's office hours. And we also act with academic integrity. So please don't expect us to just simply give you the answers to any homework, but we're of course here to support you develop those skills that you need to succeed in your classes. And finally, we also don't have access to your grades, so we won't really know which students are struggling or maybe failing. So we really recommend for you guys to take that initiative to email us and ask for help and support. That being said, the AAC is just not just here to help students who are struggling or failing. We welcome all students. So even if you're not struggling and you maybe just need a boost from B to A, we're also here to help you. We also have peer tutors that can help you. So these are just students who might have taken your courses already or are taking it with you. So if you're more comfortable working with a peer, we have 3 peer tutors with different areas of specialty, such as we have one for English and college prep, we have one in business, and we have one in science and environmental sciences. We actually are also hiring for one math or slash trades peer tutor. So if you guys are interested, please just visit the careers page on the Kano website for all the other information. And finally, one of our big initiatives again for this term is our workshop Wednesdays. So if you guys haven't heard about this yet, here's the list of all of our workshops that we'll be offering for this term. Some of them are repeats, such as note-taking, time management, midterm prep, and final prep, while some of them are new and improved, such as active reading. We also have effective presentations, how to tackle test questions for midterms or exams, or even how to use educational apps and technology, and more. So we do usually do these workshops in our center, in our study room at CC 119, but for now we're just going to do it via Zoom until further notice. This takes place every Wednesday from 12 to 12:45 p.m., although sometimes it can be shorter so that you guys don't have to worry about being late for your 1 p.m. classes. And we also usually offer free snacks and drinks for when we go back in person. So yeah, if you guys are interested to attend one, some, or maybe all of them, please feel free to click this link here, which I'll also be copy and pasting on the chat before I leave, or you can also scan this QR code right now to register. For this term, we also decided to just double our prices for our workshop attendees. So if you guys want to, maybe win a $25 gift store gift card, we will be giving away one for a random attendee and one for the student with the highest attendance. Yeah, I think that's everything for me today. Do you guys have any questions about the ASC? I'll just stop sharing my screen here and I'll just paste our general email and the link to register. Okay.
Okay, well, thanks, Dana.
All right, awesome. Well, thank you for inviting me again, Blaine, and I hope I see you guys in person or virtually.
Okay, you have a great day.
All right, thank you guys. Bye. Have a great day.
All right, so back to Biology 107. So today we're going to talk a little bit about the history of the discovery of cells and a few things around that. Cells are microscopic, so obviously we were not really able to learn much or even discover them until microscopes were actually invented. Microscopes and telescopes were invented somewhere around the year 1500, somewhere in there. And lots of people, mostly rich people, kind of got interested in them. More people were interested in astronomy than looking at small things. But this guy here, Robert Hooke, This is the same guy who did Hooke's law if you took some physics. And he had a telescope, he had a microscope, and there's a model of the microscope that he had. And he started looking at all sorts of things and wrote a book about it. And so this is one of the reasons why he's famous. Here are some of the things that he was looking at. So insects, so that's a water flea, snowflakes. And one of the things he looked at is cork. And so cork is actually a marine animal, and it's used pieces of its tissue for wine bottles and things like that. Although nowadays we're not using actual cork, we're using synthetic materials. And he looked at it and he said, "Oh, look at those little chambers in there. They kind of look like cells." Meaning he's thinking of like prison cells or cells that a monk might live in or something like that. And so this was basically the discovery of cells. And lots of people were very interested in the things he was seeing, and more people were interested in trying to obtain or get microscopes built for themselves. And so that kind of leads us to this guy here, which is a few years later. He was contemporary with Hooke. Maybe this is about 20 years later, if I remember correctly, when he started making microscopes of his own. This is Antony van Leeuwenhoek. He's a Dutchman. And he made this little microscope. And if you take a look at it, it's actually very, very simple. What you have here is a lens right there, and then a holder. And that's about it. It's in fact not much more sophisticated than a magnifying glass. There is a sample holder and these little adjustment knobs that allow the sample to be moved around closer, further away from the lens, mainly for focusing. You can see he's just using natural light to look at his specimens. And one of the things that Van Leeuwenhoek did was he started to look at things that weren't obvious to look at. You think about what Hooke was looking at, it's like, what does a fly look like? Okay, let's zoom in and look at it. He started looking at things like pond water. So this is a little video of pond water I'll play for you. And pond water is a kind of thing that, people were thinking is pure, right? What could be more pure than rainwater or pond water? And in fact, when you start to look at it now, we realize there are a lot of things in it, including microorganisms. And you saw these things swimming around, and well, they're kind of like animals, but they're small like molecules. And so the term animalcules came out of this whole thing. Van Leeuwenhoek started publishing. He started drawing diagrams of things and made, I'm not even sure, maybe hundreds of biological discoveries. You can see some of the things he discovered here. Red blood cells. No one knew that blood had cells in it until Van Leeuwenhoek. All sorts of aquatic microscopic organisms such as hydra and rotifers and things like that. He discovered that there were bacteria and organisms in his own feces. He discovered sperm, again, something that had not been discovered before and a very huge biological relevance. And he discovered bacteria. We believe he's the first person to have at least published and seen bacteria. So huge, massive discoveries here. And this kind of moves us forward a little bit to the 1700s. I'm going to skip back to the 1600s here for a second, but kind of understanding that cells are the basis of all structural biology. The problem is there was another theory going around in the 1600s called spontaneous generation. And spontaneous generation is kind of a weird historical quirk, but there were a lot of scientists actually taking this very seriously because it turns out that people could, at least it looks like they could make things following recipes. And so I'll show you a recipe and you'll get an idea of what spontaneous generation is like. So here's a recipe for making mice. You get a jar, get yourself some wheat, you get a pair of old underwear, and you store it for 21 days, and then you get mice. Apparently this does work. It attracts mice. Now here's the thing, right? We know that mice come from mommy mice and they have babies, and the whole thing seems kind of silly considering many people were farmers back then. But there was this idea that given the right circumstances, life would just basically appear. And So people were, so scientists that were studying cells were saying, well, this is ridiculous. There's a lot of microscopic things that we don't understand. And if you, in fact, wait around and watch, you'll see Mama Mouse come along and give birth to her children. And apparently this is an ideal situation for mice. So this started to get in. This is kind of the beginning of the scientific revolution, where people were starting to do experiments to either, try to confirm or prove or disprove things. And so this leads to this guy named Francesco Redi. And so there was this observation at that time that meat eventually turned into maggots. So maggots are little worm-like things. They're actually baby flies. And but we didn't know that at the time. In fact, if you look at a house fly, it looks nothing like its offspring. And so, in fact, people did not know anything about the life cycle of insects back then. Who would have known these were the same two species? So what we know now is that the fly will actually land on the raw meat, and it lays its eggs, and the eggs are microscopic, and they eventually hatch and turn into these larvae, which we call maggots. So he wanted to propose an experiment to disprove that these maggots were spontaneously appearing on the meat. And so initially he took the meat and put it in a jar and plugged it up with a piece of cork. And the skeptics were saying, well, of course there's no maggots growing on the meat because there's no fresh air getting in there. And so he changed his experiment and used a cheesecloth. That's just a cloth with holes in it that could allow air exchange, but not allow the flies to go in. And the flies actually would lay their eggs on top of the cloth, and that's where the maggots would hatch. And so this was kind of one of these experiments that was really, you could do it at home, very easy to do, and you could show that maggots don't just appear. People started to investigate the biology of flies. A bigger question, though, was what about cells? Particularly all these microscopic organisms that seem to be, people were discovering them in all sorts of places. And a lot of people were thinking, okay, these things just appear. There's no mother fly or mother mouse coming along and laying eggs. And so this actually took about another 100 years before people started to really get a better understanding of what was going on with cells and where they came from. And so Louis Pasteur, just shown here on this slide, was a French scientist, and he was really interested in food spoilage at the time, and he wanted to disprove spontaneous generation. And so what he did is he did this famous experiment called the Swan neck flask experiment, and I'll show you what's called a swan neck in a minute. You may know a swan. A swan is a big bird, kind of related to ducks and geese. So he put some media, some food in a flask, and there it is there. And you probably know what happens if you leave food sitting around your house. Eventually it's going to spoil. And his hypothesis was that microorganisms were causing the spoilage, and also that the microorganisms were found in the air, maybe on dust particles. And so his thought was that microorganisms would show up, fall into the broth, and then cause the spoilage. So what he did is he sterilized the broth by boiling it, and then he bent the glass. And so you can see here, he bent the glass into what he called a swan neck shape. Because swans have big, long, curly necks, right? So the whole idea is that air could get in. Remember, the skeptics were always worried about the air, but if dust particles fell in, then the dust particles would actually get stuck right here in the neck of the flask. And in fact, that's exactly what happened. And his flasks would not get contaminated or spoil. In fact, there is an actual flask that Louis Pasteur made, whenever that was, 100 and something years ago, and it's still actually sterile. And you could show that the bacteria were in the dust particles by you could break the flask like that, and then eventually it would spoil, or you could kind of tip the flask up to grab that dust and let the dust go back into the media, and show that that's where the cells were coming from. So this led to what we call now is the cell theory. And like I said, this is kind of the basis of all biology. All structural biology is that life is composed of cells. And this theory was formulated by these two guys, Schleiden and Swan. One of the guys was a I can't remember which was which. One was a botanist looking at plants, another was a zoologist looking at animals, and they met for lunch, and they're like, well, did you know that all plants seem to be made out of cells? And wow, that's interesting because all animals seem to be made of cells. And so that was the first tenet of what we now call as a cell theory, is that all things are made out of one or more cells. Number two is that the cell is the basic building block of structure and organization in a cell. And number three, which is relating back to Pasteur already, is that all cells come from pre-existing cells. They're not, there's no spontaneous generation going on, which we'd call biogenesis. There's been other things added to cell theory. This whole idea, for example, that the hereditary information is found in a cell, and that cells are responsible for carrying out energy functions and things like that, and so on. But really, number one, two, and three are the basis of the cell theory. Okay, so before we move on, I wanted to try a quick Kahoot. So I warned you last day that you should download the app. If you don't have the app, you can download. You can just go to the website Kahoot, and what it is it basically has some little quiz questions, and let's give it a try here. I just got to change my screen share. There we go. So here's Kahoot. What you need to do when you load up the app is you put in the PIN number there, and if you don't start right away, the PIN number should be found on the screen. And it, like I said, it's kind of like a little quiz thing, and you get to get some points and maybe some bragging rights and things like that. And so I'll just give everyone a minute to join in on the Kahoot. All right, the more the merrier. I'll give you another 30 seconds to join in. Okay, so this is just a basic one with 3 questions. And if you download the app, you can always join in anytime, and hopefully more people will get a chance to join in the next round. So 3 questions should be relatively straightforward and simple. Number 1, who discovered and named cells? Okay, so it's Robert Hooke. So Louis Pasteur, he was the one with the swan neck flasks, and Robert Hooke was the one with who saw the cork cells. So by the way, you get points, and the way the points work is the faster you answer the question, the more points you're going to get. So good job, Kyla. Number 2, what is a name? Okay, glad everyone got that right. All right, one more question. Like I said, this is just a basic one. All right, I'm glad everyone got that right, considering it's Friday afternoon. I meant to have this Kahoot last day, but fell out of sync. But anyway, like I said, just a quick introduction to Kahoots. I'll have more of them over the semester to test your knowledge. And what it does, it gives you the podium. So Emil got bronze, Izzy got a silver, and I'm guessing Kyla got the gold. So well done. I think the real question is if Kyla can hold on to her winning streak, and we'll see that next time. Yes. Okay, so I'll just go back here to the PowerPoint and continue on here. And what I want to do now is kind of transition and talk about some other things. And one of the big kind of concepts of this course is understanding, obviously, cell structures, but understanding a little bit about these 2 types of cells here. And so We call them prokaryotic and eukaryotic cells. So let's just take a look at the word here, what this means. So this is actually Greek, and it means true. And karyotic means nucleus. So that is the number one thing to know about what a eukaryotic cell is. Eukaryotic cells include animal cells, plants, fungi, algae, and protists. And I'll talk about algae and protists a little bit later, but if you don't know what those things are. Prokaryotic cells, so pro means before in Greek. And so whoever named this, I'm not sure who named it, basically threw out the concept that these things are more primitive, so that they must have existed before eukaryotic cells. And that's the idea that comes from, where it comes from, rather than saying no nucleus, they decided to call it before nucleus. I don't know if I like that definition, because obviously these things haven't changed and they seem to be doing very well. And I'm guessing they're not going to get a nucleus anytime in the near future. But anyway, that's where the words come from. And this includes both the bacteria and the archaea. And if you haven't heard of archaea, we'll talk about those as well. We'll get into what those things are exactly in the next few units. So what is a typical prokaryotic cell? No nucleus. You would think it's just a bag of liquid. It has all sorts of things. You can see it's loaded with DNA and ribosomes and cytoplasm and enzymes and things like that. It has a number of surface features. We're going to talk about what some of these things are, like the flagellum here. This is kind of like a tail used for swimming, and so on. And we'll get into lots of details on what a typical prokaryotic or a bacterial cell looks like. Here's your typical eukaryotic cell. This is actually representing an animal cell. And you can see that it also has a flagellum, and that doesn't include all animal cells. In fact, in humans, the only cell with a flagellum is sperm produced by males. But the other thing you can see are all the organelles and how much more complex this thing is. And we're going to talk about all of these things and what they do and what they're doing in the cell. So let's talk a little bit about biological classification and nomenclature. And this is kind of what I wanted to cover today. And so right now we've talked about that there's two types of cells, and these cells have subdivisions. So the prokaryotes have the bacteria and the archaea, and the eukaryotes include all those other things that I mentioned: animals, plants, fungi, algae, and protists. We are also going to talk about some things in this class that are not considered living or acellular, and that would be viruses. We're going to talk a lot about viruses. I think we're going to maybe talk 3. I'm not sure yet when we'll get into viruses, but soon enough we'll talk a lot more about viruses and COVID-19. So this is kind of another way to show the relatedness of these types of life forms. This here is actually called a phylogenetic tree. Phylogenetic tree. So phylogeny basically means how things are related. And usually in biology, what we're doing is comparing DNA sequences. And I have a little slide that shows how that might be done. And so nowadays we have all sorts of genome databases, and we can compare things. We can take a look at organisms that are plants versus fungi versus bacteria, and using various algorithms, kind of look at how related these things are. So if you take a look at the eukaryotes, for example, I've made the animals and plants and fungi a little bit larger. So you can see that animals and fungi are actually more closely related than plants. We have a lot of similarities in terms of our metabolism. We eat things. Fungi eat things, although a lot are decomposers. But from a cellular level, there's a lot of similarities compared to a plant, where plants are typically photosynthetic and have chloroplasts and all those kind of things. And there's all sorts of other groupings in there. We kind of get away from using the word kingdom nowadays, because I think there's a lot of debate in terms of how to classify these things. You can see the bacteria and the archaea are classified in their own groups. And so another way to classify living organisms is 3 domains, meaning that all 3 of these groups are different enough to be considered their own thing. Remember, though, archaea and bacteria are prokaryotes, they have no nucleus, but they are different enough, and we will talk about what those differences are a little bit later on in this course, topic 3 or something like that. So there's the 3 domains of life, some pictures of them. You can see some bacteria and archaea. They're microscopic organisms, and then our protists and our animals and fungi. So what is a protist? Let me just talk about that for a minute. You can see this protist here that looks like some sort of amoeba. And so a protist is a eukaryote. Remember that it has a nucleus, and it is usually unicellular. Not always, there are some exceptions. And it doesn't fit into one of the other categories. So it's sort of a weird catch-all, meaning it's not an animal, plant, or a fungi, but it is a eukaryote. So that's kind of what to know about protists. They're eukaryotes, usually unicellular, but not a fungi, an animal, or plant. There's a huge amount of diversity, of course, in the protists, and we'll kind of talk a little bit about that here and there during the semester about protists, and we'll hopefully get a chance to look at some protists in lab 5 in this course, which is very cool. So let's talk a little bit about classification. I was just talking about phylogeny and how we look at relatedness of things, and I know I have a slide coming up showing how that's done with the DNA. You're probably more familiar with taxonomy. So taxonomy means classification based on physical traits. So let's take a look at an example. So here's a human, and human is part of the eukaryotes because we have a nucleus. So that's which domain we're in. We are animals. Animals are heterotrophs or multicellular, meaning we have lots of cells. Heterotroph means we feed to eat. And usually most animals, this is not all the criteria to define an animal, by the way. There's only so much space on a slide, but usually we say animals are mobile, meaning we can move around, right? We are in the chordata phylum, which means we have a central nervous cord. We are mammals, we have hair, we give milk to our young. We're also primates, which means we have opposable thumbs. And if you look at a lot of other mammals, dogs or cats or cows, they don't have these opposable thumbs. So that puts us into the primate category. There is another type of opposable thumbs that's found in pandas and raccoons, but if you look at the bone structure, it's actually quite a bit different. So they're in their own order. We are hominids. So we have no tail. There are primates with tails. So technically we'd be considered apes. We have a large brain case that also makes us our own thing, because I think a lot of other apes are in their own category, but I'm not entirely sure about that. And then genus, we're Homo. Homo means human. And I always ask people what they think species means. Species, everyone knows that we are Homo sapiens, but what does that mean? And here's the thing, if you get to name yourself, you get to be flattering. And so sapiens means wise or thinking or sentient or something along that line. So this is kind of how we classify things. And I'll give you another example in a moment. And I know a lot of people get really bogged down and worried about all of this stuff. But these are the 3 things I really care about. Care about domain. Is it a eukaryote, a prokaryote, a bacteria, or an archaea? That's kind of what I care about. And then genus and species. And I'll talk about genus and species in a few minutes in terms of why that is important for this class. So let's take a look at another example. Let's take a look at a dog. You probably know that a dog is a mammal. And so in its classification scheme, it is exactly the same as humans, all the way down to that classification of mammal. So if you remember that mammals, humans are primates with our opposable thumbs. Dogs do not have opposable thumbs. In fact, they fit into a group of animals called carnivores. And a lot of that is based on their teeth structure, not necessarily their diet. Because a dog is not a complete carnivore, by the way. A lot of people don't know that. But dogs are actually omnivores. There's actually quite a bit of plant in their diet. If you take a look at the ingredients of your dog food, if you have a dog. Cats, on the other hand, are almost strict carnivores. They almost eat exclusively meat. So the family, they're in the canines, which literally means dog-like. So that includes foxes and coyotes and all sorts of things like that. So genus and species, Canis, and the species is Lupus. So lupus means wolf. So that's right. Dogs and wolves are in fact the same species, and they can breed with one another. And in fact, some dogs look a lot like wolves. If you've ever seen a husky, it's very wolf-like in appearance and whatnot. Domesticated dogs, of course, there's many interesting, weird, and wonderful varieties. So usually at this point, I ask people if anyone's ever heard of that book series or movie series called Harry Potter. And if you remember the third book movie, there's Professor Lupin. So Lupin, of course, means wolf. And you find out much later on in the book that Professor Lupin is in fact a werewolf. So you knew that what lupus means, and you would have known that there was a clue there right from the very beginning. So one other thing I wanted to show you before I move on is that sometimes we will go further and we will actually talk about subspecies. So a lot of organisms, there are different varieties, and the domesticated dog is actually it's considered a subspecies of the wolf. So it's Canis lupus familiaris, and familiaris means friendly. So sometimes we classify things a little bit further than the species. So let me show you another classification scheme for something that you're probably less familiar with and is totally different. This is a bacterial organism, and this is we're going to use taxonomy to basically do the same thing, which is going to be a little bit more difficult when we're looking at microorganisms, and you'll see why this is in a moment. So bacteria, remember, they're prokaryotes. They have no nucleus, and if you want to distinguish them from the archaea, they have unbranched phospholipids. So we're starting to get into some chemistry here now. This particular group is gram-positive. We're going to talk about cell walls soon. This cell wall is thick, and so that fits into its own kingdom. They are firmicutes. Again, now we're starting to look at some molecular stuff in terms of low G and C versus A and T in their genome. So that's kind of interesting and weird. The next two are based on genetic sequences, at least they are now. Historically, I'm not sure what they were based on, but actually we're doing that now. So here's where we're getting back into characteristics. If you take a look at that particular organism, notice that the organism is, these are spheres. And so coccus means sphere. They're also found in clusters. It looks like a bunch of grapes, and cluster is staph. And so that's actually the genus as well, and we call this Staphylococcus. So we're going to see Staphylococcus a whole bunch of times this semester. We're going to look at it in the lab and all that. And this organism always looks like this, right? It's spherical in shape and found in its clusters. This particular species of Staphylococcus is Staphylococcus epidermidis. And you probably know that epidermis lives on your skin. I think I put in some extra letters in there. I misspelled that. I'll have to fix that PowerPoint. But it lives on your skin. Your skin is epidermis. So as I mentioned, it gets a little more difficult with some organisms to try to classify them. And it's worth kind of mentioning what we do actually mean by species. And the kind of the historical, traditional biological definition of species is things that can breed together. So I mentioned dogs and wolves are the same species in that they can breed, and you get baby wolves or baby dogs or whatever you want to call them. Whereas if you take a dog and a cat, they cannot breed with one another. Sometimes there are some weird organisms out there that do breed together. So if you take a male donkey and a female horse, you might know that you get a mule. So a mule is not actually considered a species. Mules themselves cannot breed. You can't have two mules. They don't get together and make baby mules. It is a living organism, yes, but it's not considered its own species. It's considered a sterile hybrid. Believe it or not, there's a whole bunch of these things out there. You can look them up on the internet. Here's another one, a male zebra and a female horse. Some people are probably wondering, what do we get? Do we get a zorse or a hebra? I don't know if I actually have an answer for that. Maybe it says there, it's a zorse. Okay, it's a zorse, of course. Like I said, you can look these up on the internet. And again, it's the same thing. This is actually these two are not compatible in terms of making a viable offspring. It's actually a sterile hybrid as well. So like I said, when we get away from animals and we start to get to microorganisms and even plants, and sometimes things get really, really complex, particularly bacteria in general. It's not like there's a male and female. They don't even breed. They reproduce asexually. Sometimes they swap DNA and they don't even have to be related. And so sometimes we need another way to do this. And so a lot of organisms now, we get away from taxonomy and we move towards phylogeny. And so remember, the taxonomy is characteristics, physical or chemical characteristics, and phylogeny is relatedness, where we're often looking at DNA sequences. And in fact, usually what we look at is ribosomal RNA sequences. All organisms have ribosomes, so it's kind of a good basis for comparison. Or to look at human genes, let's say the gene for, I don't know, eye color. How is that useful when we're looking at plants? Plants don't even have eyes. So ribosomes everyone has, so it's kind of a good basis for comparison. So we'll be talking a little bit about some of this stuff in Lab 4. Just want to show you kind of how we can sort these things out in terms of the relatedness and phylogeny. You can see I have a couple of examples there of two organisms, just a very short sequence of nucleotides, 20 nucleotides. But you can see that if we look at organism 1 and 2, and this should say 3, oops, 1 and 2, they're more closely related. There's only 3 differences. So in that case, they're like 85% identical. So they're more closely related than organism 1 and 3, which you can see there's a lot less common base pairs. And so this is how it's done. It's a little more complex with computers, and you're looking at thousands and millions of base pairs, but basically that's how it's done. So back to our 3 domains of life, just wanted to point out this little thing I have at the bottom here that says they are sorted often by looking at DNA sequences. Here's a kind of a more modern publication of somebody else looking at all of the different sequences in the database. And this person here actually puts the archaea and the eukaryotes a lot closer than they do the bacteria. And what they found was there's a huge amount of diversity in the bacteria compared to the archaea and eukaryotes as well, which is very, very interesting. I'm just looking at the time here, and I want to talk about nomenclature, but I think I'll get into most of this next day. But I do want to kind of just talk about it for a minute, and then we'll come back to this next day. I mentioned that I was interested in genus and species, okay? And so if you take a look at this, how this is done is genus and species are usually some sort of Latin or ancient Greek word, or sometimes there's a few other ways to name things. The whole idea is that there's no favoritism, right? We don't want to use dog because that's a human word. And then of course the French people or the Germans or whoever is going to be upset that they're using the English word. So this is why we use dead languages. There's no favoritism. So sometimes we call these the Latin names. The technical name is binomial nomenclature system. So binomial means 2, right? 2 names. And we give these things 2 names, a genus and a species. And the last thing I want to say is that it's important you get this right for lab reports. And I'll come back to this and talk about it next day, that this is capitalized for genus, lowercase for species, and the whole thing is italicized, or if it's handwritten, you can underline. I have a lot more to say about nomenclature. As you can see on my next slide, I'm going to give you some examples of how that works, but I'm not going to worry about that today. We'll come back to it on Monday. This is where I'm going to finish for today, and then we'll pick this up where we left off on Monday.